All right, I think that's been enough time. People can join in. So before I introduce everybody, I do want to thank everybody that is currently attending, and for those of you that are watching the recording later, hello, and thank you very much for being here. So my name is Brett McAllister. I'm the Director of Graduate Admissions for the Halmos College of Arts and Sciences. With me, as most of the time, are two members of my staff, Ms. Farah Amro, and Mr. Theodore Uwe, who most of you marine science students have been communicating with or will soon do in the future. Um, Now, this is the Halmos College of Arts and Sciences virtual lecture series, with our focus tonight being our brand new department chair, who I am very thrilled to be able to introduce, Dr. J.R. Kerfoot. And if you excuse me, I'm going to be reading from a bio over here to make sure I don't mispronounce anything. So the title of today's lecture is Don't Tempt the Gators and Other Things I've Learned Along the Way. Now, Dr. Kerfoot earned his Bachelor and Master of Science degrees at Southern Illinois University in Edwardsville, studying molecular transformation techniques in diatoms and then ecomorphological studies of the banded sculpin. He earned his Ph.D. from Florida Institute of Technology, investigating the environmental dispersion and spread of the non-native pike killifish in South Florida, along with its feeding morphology and disruptions in the trophic dynamics of the Everglades. Now, before joining NSU in June of 2025, Dr. Kerfoot has had a disparate teaching and research career that includes courses from human cadaver-based labs to tropical ecology to wildlife management and research topics that span from harmful algae blooms to alligators to pitching kinematics to seagrasses. So I hope I didn't mispronounce too many of those words, Dr. Kerfoot. Thank you so much for being here. With that, I will turn it on over to you. Also, one last quick thing for those of you that are watching. We will be doing questions at the end of it that will be typed into the chat, but we will speak with you about that a little bit later. So now, Dr. Kerfoot, the floor is yours. Thanks for being here.
I appreciate that, Brett. I need to go ahead and share the talk, so I'm going to go ahead and do that now. Here we go. Give me a hearty hey yeah if you could see that.
There we go.
All right. And so thank you, Brett, and thank you, team, graduate admissions team, for your welcome here. And so this is kind of interesting. I just started at the end of June this year, and I'm so blessed to be here at Nova Southeastern University in the Department of Marine and Environmental Sciences group. And it's kind of a coming full circle for me. And so I want to talk a little bit about my journey. And it does involve Florida, but it didn't start there. And so I love this picture because it's me down in the Everglades for my PhD project. And you never go to the Everglades when you're fishing at night alone. And that's because you have these two little eyes right here. I don't know if you can see that. I don't know if it's picking up on your screens, but that's an alligator. And so we had to contend with alligators as I was trying to find my little fish that I was doing my research project on. So again, never go down to the Everglades alone, especially at night when you're trying to fish. And so how does that work into just what I've learned along the way? Well, I really have no special talent. I'm only passionately curious. And I love this quote from Albert Einstein, because growing up, I was always curious about everything and asking questions about how things worked, why things worked the way they did. And so I think that's probably a lot of what you guys are about. You guys are interested in continuing your education and asking, maybe addressing those types of questions, why and how. And so those were two prominent questions growing up. I always liked to tinker with things. And so I think that's really what's driven my curiosity all along. And so you see this on the screen. To a lot of people, you might not know what that is, but others that are more astute, hey, that looks exactly like a gear. And so I would see something like this and say, well, how is it working? And we know that gears have teeth, that they can fit together and move and articulate against one another to help cause an effect. And so, again, these types of questions interest me. And if we kind of back out, we actually see this mechanism here is from a plant hopper. And it's situated on the edges of the legs. And it actually helps in propelling this plant hopper Actually, it propels them about to 400 Gs, and that's 20 times greater than what humans can sustain. And so, I mean, now granted, it's not a very far distance, but it's in a rapid speed. And so how does it let this guy work? Well, he can escape from predation. These guys are going to be food items for a lot of things if it didn't have this crazy ability to escape in this way. And so, these kind of things I would see in nature, or I'd see in man-made things and ask questions of how do they work? I want to know more about it. And I just sit there and tinker. And so these kind of questions interested me growing up. And I grew up all over the place. I'm an Army brat and born overseas, graduated high school overseas, but spent lots of time in the United States as well. And just this curiosity leading me left, right, and center. And so as we think about the journey, it took me through, of course, undergraduate, but really what I wanted to talk about tonight was my graduate journey and how it got me to where I'm at today here at NSU. And so I'm really interested in a lot of different things, but I used to take a look at animals and ask the question, why were they in this habitat? What makes them adapted for these types of habitats? And so really it plays on this idea and this interface between morphology and ecology. And we know that in some cases, small changes in morphology can lead to novel functions, just like the plant hopper, just those gears being able to really articulate and focus that propulsion in one direction allows those plant hoppers to do some pretty crazy, wicked things. And so that's a small change that has a really novel function. We know in fishes, so I got interested in fishes. Going from my undergrad, I did a lot of molecular techniques on diatoms and that sort of thing. But then I decided, hey, I wanted to go outside and do some fish. And so we know that fish body forms correlate pretty well with the microhabitats that they're found in. And so what's interesting about that is their morphology adapts them or aids them in their environment, in their ecology. And so I was interested in questions that related to that. And that brought me to Cottis carolina. It's also known as the banded sculpin. And so sculpin are pretty neat because they're just bottom dwellers. They hang out along the surface of the substrate, and they're going to eat things that are on the surface of the substrate. They don't really tend to swim up in the water column or anything like that. And so because they're up against the substrate, they have seemingly adaptations that allow them to maintain a stationary holding ability on that substrate. And so you see kind of these big pectoral fins on the picture here, this cartoonish picture, and really a long, slender, tapered tail. Those are adaptations that can allow these guys to be, in a sense, more hydrodynamic, or at least where the water can flow over them and not pick them up and wash them away. So it helps in really maintaining or conserving that energy as they kind of just maintain that holding position. But what was interesting is in part of my beginning of my master's program at Southern Illinois University, Edwardsville, was going out and kind of just surveying different types of streams, some prairie streams and some Ozark streams. And if you were out there in the same streams surveying them along with me, you'd see a lot of things that were different about the streams. In the prairie stream, they're slower flowing, they're very silty and muddy, and it's so they don't have a lot of rocks or rubble in the substrate. However, Ozarks, and we're talking about in Missouri, that sort of thing, those Ozark streams are faster flowing. They have lots of rubble and gravel and cobble stones, and they have lots of riffles and that sort of thing. But the interesting part in my mind was that this species of fish occurred in both types. And so I asked the question, does the morphology change based on where it's found? And so again, I have these two different locations. And when I think about research projects, I'm always interested in this kind of a dichotomy. If I can find that there's something different about maybe locations or temperatures or something like that, and in this case, the streams were completely different, can I see if the morphology goes along with that change or that difference? And so I took 21 morphological measurements were made, all sorts and manners of different types of measurements, the width of the head, the depth of the head, The caudal peduncle, you might not know what that is, but that's in this area in the back of the tail, looking at the width of it, looking how big the pectoral fins are, the pelvic fins, all sorts of things, the length of the body. All these might have ramifications for its ability to remain stationary in a flowing body of water. And so I was interested in that. And again, this is only part of the study, but what came out of that study, and this might be a hard graph to kind of explain or understand if you've never seen one before, but what I want to focus your attention on is on the y-axis, we have this measurement of velocity. That's the water velocity. How fast is the water flowing? And so all you really need to know is that it can go from 0 to increasing in velocity, okay? And so it's faster flowing as you go to the top. Along the x-axis, these are just different, those 21 morphological measurements. The ones that are actually outlined here, like the head width, the head depth, body width, and all that kind of things, those are the most important measurements that I took according to the mathematical model that I used. And what we notice here, that the same species, again, these are the same banded sculpin from 2 locations. In blue, you see that's the faster flowing stream, right? And in red is the prairie stream that was the slower flowing and had the silty, muddy kind of substrate. And what we see is those that are in the slower flowing stream had bigger heads. Well, I guess that kind of makes sense. If the stream is flowing slower, they don't need to be as hydrodynamic. However, those that were in the faster flowing stream, in the Ozark stream, they had narrower heads. They had the depths of the heads were not as great. And so this allowed them to remain stationary even at higher flows. And so another part of the study that I don't show the data for actually include taking some live specimens into the lab and from different locations and ramping up the velocity of the water, and the same pattern emerged. You had those prairie banded sculpin didn't hold as stationary ability in the faster flowing streams. And so this was one of my first papers published, and I was so excited to get it published. And I tell you what, though, as you'll find out along your journey, your writing is going to get better and better the more you practice it. And so I remember as a story here, my major professor, Dr. Schaefer, Jacob Schaefer, he didn't really proofread my manuscript at all. He said, just send it in. Well, I got it back and it was tore to shreds, and he just laughed. I said, why didn't you help me out and read it in the first place? He said, the greatest teacher is going to be those editors. He wasn't wrong. And so that's another lesson that you should take from this. Good writing is rewriting every time. Well, so the idea of morphology interested me a lot. And to take kind of a step back, I went for my master's in Illinois, and I wanted to pursue a Ph.D., but I was really interested in looking at the feeding kinematics in sharks. I didn't end up working on sharks. That's another lesson maybe that I can parlay to you guys, is that you have a plan of what you'd like to do, but there are so many different routes you could go and that aren't wrong. And so I wanted to look at shark feeding. I ended up looking at the feeding kinematics in pike killifish. And this is a pike killifish here. It doesn't quite have the same kind of dental structure as a shark, but it still has a pretty fascinating one at that. And it's probably 4,000 times smaller than a shark. So it's going to be this little guy that I'm interested in. But I was continuing this idea of this ecomorphology. How does the morphology relate to a fish species in this case? But then I added something else, and I want to talk a little bit more about that. So I went into the PhD program at Florida Tech, and so up in Melbourne, Florida, Florida Institute of Technology. It's called Florida Tech now. I think the institute is still there. But I studied under Dr. Ralph Turingen, who's a functional morphologist and is big on feeding kinematics of fishes. And that's just a fancy term for thinking about how the mouth moves and grooves as it collects prey or captures prey and that sort of thing. And there's a huge diversity, even in fishes, of how that's done. And so now, but what interested me about pike killifish is that it's also known as an invasive species. It was introduced to South Florida. And so that, again, thinking about well, what type of tangent can I actually apply to this introduced species? Well, the introduction of species, as it outlines here, is the opportunity to study the processes that structure the distribution and abundance of species in its novel environment. And so I love this picture because it kind of gives us an idea of how an introduced species becomes established, or really introduced, then established, and then it can spread in the environment. And you see at each of these stages, we have these invasion stages: the transport, the establishment, and spread. We know that not everything that gets transported, to a new area can survive that transport process, whether it's in the bilge of a ship where larval fish get sucked in, and then in a new location, as that bilge is spread out into the new area, those larvae become introduced now into that area, but they might not survive. They might not have the tolerance to do that. But if they can tolerate that new area, they become, quote, introduced. Well, then it moves into this next stage, the establishment stage. And what's interesting about the establishment stage is that requires that they actually produce they actually produce a population that's self-sustaining. So they have to find a mate in most of the cases that we're going to talk about, at least in fishes, right? And so they have to find a male and female and make babies. And so what's interesting about that is not every introduced species, any species that can be introduced, actually moves past that establishment stage because they haven't made a self-sustaining population. But if they can, then they can move into this next stage called the spread stage. And that's where they move out from their initial point of introduction into kind of these heterogeneous environments away from it. And what's interesting about that is that heterogeneous environment can pose different stressors on that fish, and that's fish species. So what's interesting about things that can affect or regulate spread, they can be abiotic. So we're talking about weather phenomena, temperature, dissolved oxygen, pH of the water in the case of the fishes. But you could also talk about other factors that are biotic. So competition for food resources from other species, or competition for food resources maybe they're limiting within your own species, competition for mates, those sorts of things. And so there's really this dichotomy of abiotic versus biotic factors that can really help regulate the spread stage of these invasive species. And really, if we're being honest, it's actually a combination of both. Now, for this study, I was interested in looking at the abiotic factors, but I got into a lot of the biotic things because that's really where the interesting stuff is also. And how maybe these abiotic factors affect those biotic, maybe competition and that sort of thing. And so really the physiological and behavioral and ecological potential of the invasive species really regulates if they are successful or not, but it's also the properties of the invaded ecosystem. We have deserts, they are not very good at being hospitable for invasive species. It's just a really harsh environment. Whereas we have marshlands, they're pretty good actually for invasive species. And so as we think about that as it relates to the pike killifish, again, these guys, part of their name, Belanisox belizanus, really points to where they're from, Belize. And so they're actually from Central America in more than just Belize, but they were first described in Belize. And the anus part actually has an important meaning because it is a live bearer. The males have this gonopodum that comes off the anus, and that helps to inseminate the females. And so if you're wondering, that is a weird name. That's why it's a weird name. Now, how did it get to Florida? Well, the story goes, we can blame a lot of things on the University of Miami. No, I'm just kidding. But they had a medical school in the University of Miami back in the late 50s that actually used this fish because it's a live bearer. It gives birth to little miniature versions of itself. And so it doesn't go through kind of a larval stage necessarily. It gives birth to it has all the form of an adult, it's just not as big. And so because it has all the development of the skeletal and muscular system, they're great for thinking about medical kind of lab rats, as it were. But in the late 50s, the story goes, the University of Miami ran out of funding for this research that these guys were being used on. And so what do you think happened? Well, all those fish, instead of being euthanized, they were poured into a Dade County ditch, Miami-Dade County ditch. Well, they were first then cited and reported in the Everglades in the 80s. And so, again, it took some time, but we all know, especially if you're from down here, that all canals kind of lead to the Everglades. And so the pike killifish made its way to the Everglades from 58 to the 80s is when it was announced or noticed in the Everglades. But then by the 90s, it was actually noticed in the northernmost part in the Big Cypress National Preserve, sorry, Big Cypress National Preserve here. And that's kind of where it remains here today, even though there are probably some canals along the East Coast that it's also increasing. So what we have here is kind of interesting. It is a movement from a southern portion to a northern portion. Now, I get it. The climate is not so different, but then that got me thinking about how it's spreading and what to do about it. Now, long story short here, there was a lot. This was part of my dissertation and only one little small part of it, but I was looking for what abiotic factors affected the densities, the population numbers of this fish in the Everglades and in Big Cypress National Preserve. And so I had lots of help from lifelong researchers down in the Everglades that shared their abundance data with me over different time periods. And we also had then environmental parameters like dissolved oxygen, temperature, pH that were included in those data sets. And basically, I ran a big modeling program that looked at which variable or combination of variables was most important at dictating the increase or decrease in the population sizes of this fish, right? And in different locations. And that's what you see over here in this table 5. You have the Rocky Glades, Southern Red Mangrove, and then TSL, SRS, and WCA. These are different locations within the Everglades Park. But what you notice are those first lines where we have pH, then we have this T and then star S. That just means that's an interaction between temperature and salinity. And then area was included in that. And so why do I say all this? Those 3 locations had models that predicted the best population numbers using those variables. So basically pH was important, and that's what you see here in figure 3. Densities increase as the pH increases. And again, it's pretty messy. That it looks like a shotgun went off on my graph, so it's not very clean and tight. But again, this is a modeling approach to try to find, can we tease out anything that might be relatively important? And then if you see in figure 4, we actually have the interaction between temperature on the x-axis and salinity on the y-axis. And the circles here represent, larger circles represent greater population densities for this species, and smaller circles are less. And you do kind of see that there tends to be a sweet spot of roughly 23 degrees Celsius and low salinity. So you're talking about brackish water to fresh water is probably the sweet spot for where you're going to find this pike killifish. And so that led to a second part of the dissertation study. Once we started teasing apart what might be really important abiotic factors, led us to really focus a bit more on temperature, because temperature is a lot easier to control in the laboratory setting. And so from that, we know temperature for fishes in particular can influence a whole mess of things. And why is that? Well, fish are poikilothermic. That just means that the old adage is they're cold-blooded, right? So they, their internal body tends to be more regulated by the external temperature. And so metabolic processes and that sort of thing. You see that even as our list here. But all these other things can be affected, the abundance distribution, the swimming speed, growth rate. And so in the lab that I was in under Dr. Turingen at Florida Tech, he was very big into performance parameters. And so that led us to thinking about how temperature can influence the pike killifish in different ways. One of the ways that we didn't know is how it might affect the different life history stages for this species in general. There was some data out on how the temperature affects the adults, but nothing about earlier life history stages. And so that led to thinking about questions: how does temperature tolerance for pike killifish vary with age? And so we had, again, this is a live bearer, and so it gave birth to miniature versions of itself. They are carnivorous at neonates just like they are at adults. And so it's pretty fascinating to see. And so the question was, do the thermal tolerances change across each of these life history stages? And at the moment or temperature at which they cease to feed, is that different at these life history stages? And why is that important? Well, I could say, okay, they're tolerant to this degree or that degree, but really, if it matters if they are eating or not, because if you don't eat, you don't survive anyway. Maybe the more important question is, what temperature do they cease to feed? Because they can sustain themselves for a bit longer, but it's the temperature at which they stop eating that might be the most important thing. And so I was interested in the life history stage difference, but I was also interested because I knew that the initial point of introduction was south portion of the Everglades, and their most northern range at the time that I was studying them was northern Big Cypress National Preserve. And so I wanted to know, are there tolerance ranges different based on those locations also? And do the temperatures at which they cease to feed, are they different as well? And so again, sometimes you do experiments and research just because the answer is not blatantly obvious, even though you might say, well, that's a short distance. There probably is nothing different, but do we really know? And so that also is a question that plays in my mind. Do we really know? Are we sure? So what you see here are some of the results from the temperature tolerance study. What I want to draw your attention to, of course, is the x-axis, which is the acclimation temperature. There have been lots of studies that show that thermal tolerance studies are heavily influenced by the temperatures at which individuals are acclimated at first. So if you want to get a true picture, you need to have multiple acclimations for these fish, in this case, to then actually get a true estimate of what their temperature tolerance would be. So acclimation temperature along the x-axis, and along the y-axis, you actually have the lethal minimum temperature and lethal, what point they died, what temperature they died. So that's the lower range. And the lethal maximum, what temperature maximally did they perish at as well. And you have adults in circles, juveniles in squares, and neonates in the filled triangles. And what I want to, how you might read this is, okay, they all look pretty close. And looking at the lethal minimum temperature, you run the statistics and you're like, okay, well, interestingly enough, acclimation does play a pivotal role here. You see that there's really some difference at the acclimation of 30 degrees Celsius. Okay, well, that's not quite surprising because we actually see it on our picture here. But also life history stages have differences in their lower lethal tolerance. And that's what you see here, where the juveniles actually have the lowest lethal tolerance or lethal limit. Whereas the neonates, maybe not so surprising because that kind of makes sense. They're smaller and their bodies maybe can't sustain the lowest lethal temperature. And so they're, about 13 degrees. And then we have maybe the adults are about 11. And then we have the neonates around 9 degrees. And so that was kind of surprising. And so again, the juvenile life history stage was really driving that big difference between the three life history stages. But next you could look at maybe, and there was no difference in the lethal maximum, by the way. You can probably kind of see that from our graphic here, is that they're all pretty much the same. They all die about the same heat. And that actually is not so surprising in a lot of different temperature tolerance studies. That's actually been shown to be true. There's just a point at which proteins in the body of the fish break down, and it doesn't matter if you're an adult or a neonate, you're just done for at that point. another way to look at these kind of lethal minimum and lethal maximum temperature challenges are looking at these thermal polygons. And thermal polygons, just like it implies, if we're in geometry class, a polygon is a multi-sided shape. And so we can actually take the area of these and compare each of the 3 different life history stages. And what we see here is that the juvenile thermal polygon here, again, they're the ones with the squares. If you take the area there, they have a greater area than either the adult and neonate. And and so what we know from this is that the juveniles tend to have a higher tolerance overall than the other 2 life history groups. Now, again, I told you the cessation of feeding or the no feeding point. We wanted to, in this case, I wanted to compare the juveniles and adults because they fed basically on the same, they're strictly piscivores at a certain size. Whereas the neonates, they'll eat invertebrate prey and that sort of thing. But adults and juveniles, I wanted to look at the same type of meal, and so they're both piscivores. And what we notice is something kind of surprising. There is, looking at life history stage, there was almost a significant difference. If we're going to be true to our statistical textbooks, if the cutoff is a p-value 0.05, it's almost there, right? It's not less than that. But if we were to take a 1 away, yes, it is significantly different. But what we notice is the lower cessation of feeding temperature for the juveniles. Again, the juveniles are squares. At the highest acclimation temperature, they cease to feed roughly just above 13 degrees Celsius. Whereas looking at the adults, look at that. They're about 15.5 degrees Celsius. So roughly 2-degree difference between juveniles and adults. Now, again, why is this important in my head? It's because if they cease to feed, they cease to live, period. And so if juveniles can still feed at 14 degrees, they can survive, but adults can't. So why might that be important? Could the juveniles be the dispersion mechanism, this dispersal kind of life history stage? Perhaps. And so that's the curiosity that came about. Now we can also look at the data in a different way, looking at the northern adults and the southern adults. And what you see here, there's really no difference in their lethal minimum and maximums. You see the acclimation temperature is different, but really looking at, there is a subtle difference, statistical difference between individuals from the north. Those are those in the Big Cypress National Preserve, their northernmost range. In their lethal minimum, they tended to be a little bit lower. Again, it's statistically different, but it's kind of hard to see from this picture. But really, the important thing to me, again, is looking at that cessation of feeding. And so their thermal polygons were not different, but how about the feeding? Here's where it got really interesting. And so looking at the lower lethal minimum, when they stopped feeding, northern individuals in dark circles had lower lethal cessation of feeding, about 13.5, compared to around 15.5 for southern adults. And these are just adults here. And so that's interesting. So now we think, okay, juveniles have a lower cessation of feeding, but now northern individuals have a lower cessation of feeding. And so both of these kind of ideas came out of those studies. And again, this most recent study relating to the north and south, are the northern individuals gaining some sort of thermal adaptation, gaining a cold tolerance? I don't know. I want to continue this. I want to see. We also know that there is a population of pike killifish in a separate introduction up in Tampa Bay. And so I'm hoping that they still might be there. I don't know if it's an established population, but there was a separate introduction and there were reports. And this is, again, back in 2008. So it's been some time. But up in Tampa, that would be a really cool outgroup to add to this narrative to see if they are gaining even cooler tolerance. Because the idea is southern Florida never freezes. The frost line, though, Melbourne, it comes down to just north of Lake Okeechobee. And so we know that it freezes at least once a year. And so that's just kind of this idea, how far north can these guys go? And these studies helped to try to kind of tease that apart. All right, so then to completely change everything, and I've done lots of things in between, I actually took a position at Union University in Jackson, Tennessee, right after my PhD at Florida Tech. And I never sought out to do alligator research, but I tell you what, there's something very interesting on how things happen and how things play out. So another lesson I've learned is just be open to anything. Now, I'm going to tell you a story about how other students have influenced my own curiosities. And so I met a student. His name was Micah Fern. And I was just starting at Union University in 2009. And he comes to me roughly in fall 2010. And as an undergrad, at that institution, you had to actually do research projects. So nobody could graduate unless you did a research project where you're paired with a faculty that shared your interest in that research project. And actually, he was kind of funny. He said, I really want to work on alligators. And we're in West Tennessee, folks. We are not anywhere near alligators. And I just had in my head, okay, where am I going to put alligators that are, 10, 12 foot long? And I said, there is no way, there's no way. And I literally said, you are nuts. And, I thought kind of that was the end of it. But he came to me later, after thinking about it for a couple weeks, and he said, Dr. Kerfoot, what are you doing for spring break? I said, I don't know. What are you doing for spring break? He said, I'm going down to coastal Louisiana. You need to come with me. I found a hatchery. And so in spring 2011, we go down to this hatchery that I'll talk about a little bit more. And we're sitting there doing alligator research at this hatchery, never thinking that was even a possibility. But Micah Fern's curiosity was so strong, and he wasn't going to take no for an answer. And so if you're so curious about something, try to make it work in a way that maybe sometimes is outside of the box. I would have never thought about going down to an alligator hatchery and doing the research, but he did. He put us into contact. I still thought he was nuts, but he took us to the Rockefeller Wildlife Refuge. And if you've never seen an alligator hatchery, basically what this refuge does in coastal Louisiana, they actually go out and they find alligator nests, they take eggs out of there and they take it to the hatchery. And the idea is that they're going to grow them, hatch them. So the success of reintroduction after they've hatched and are raised a little bit. And so this helps hopefully curb nest predation, that sort of thing, down in coastal Louisiana because it's an important species. And so we went down and we were given access to these holding cages. And what's really interesting is his project, really, he also made a connection with a gator outfit in Florida that does kind of these tourist attractions. And so he was able to get skulls and stuff from this gator outfit. And so we actually designed an experiment that looked at changes in morphology. You've seen this here, remember? Back from a study earlier that I talked about, small changes in morphology or simple changes in size can lead to novel functions and performance consequences. What's interesting is we can start out here with a hatchling that's 95 grams, and over the course of this guy's life, it grows 4,000-fold, 4,000-fold to an adult. But there's lots of changes in and throughout. And so what this study sought to do, I'm going to show you guys this video in a minute, but what we wanted to do is look at how the skeletal structures change over time of the feeding apparatus. All right, but then we also wanted to look at the performance aspect of the feeding apparatus as it changes from small to large. And so we were able to get some skulls from the Florida outfit, and then we were able to get some smaller skulls from the hatchery, take our morphological measurements, kind of like what we did for the sculpin, but a lot of, there's some nuances with alligator skulls. And so we were able to do that and look at that. And then we were able to get in with these alligators. So this is a scale bar we had a high-speed camera, and we were just looking at how the mouth moves and grooves on these guys. And we were taking measurements, and then we were able to slow down the videos and take what we call kind of kinematic variables, like how fast they open their mouth. What is the articulation of the skull as it relates the cranium to the vertebrae? And we were looking at angles, we were looking at speeds, duration of the feeding event, and the like. And so what's interesting and what I want to point out here, these guys are hatchery raised at this point, and they were only fed kind of a kibble, a pellet food, specifically manufactured for high protein diet for these guys. And so I didn't know that coming into this. I'd never really done this, but I figured, oh, they would like chicken. And so this is a piece of chicken suspended by a fishing line that you can't see. And so it actually took a pretty good hour and a half for these guys to get the taste of chicken in their mouth because they weren't going after the bait, as it were. But once they tasted chicken, it's almost like they told their buddies. And we started getting videos of flocks of these guys coming after this. Just fascinating to think about. But what you're seeing here is a video of this guy feeding. So he misses the first time, he gets it the second time, and then I just that gut instinct, I start pulling up the fishing wire, and I didn't mean to pull him out of the water, but you see him kind of thrashing there. But again, we can go back and look in slow-mo and start these types of measurements, looking at how far the mandible is from the maxilla, so maybe a max length here. That gives us an idea of how they're moving their muscles to capture a prey item. And so we did a series of videos on that kind of thing. And our study was the first one to really reveal that feeding performance scales allometrically with body size. They kind of already knew that with the limbs and appendages, because if you're living as a hatchling on the terrestrial systems, you're going to be then, as you grow in size, you need to take some of that weight off. And so going into the water helps alleviate some of that stress on those limbs. And the limbs get stouter, and they're able to move a lot better in water than, say, lumber around on the terrestrial system. As alligators grow, though, in our feeding study part, you see that the increase in force production rather than the velocity and timing. What does that mean? They actually are very similar as a small individual eating, doing the timing and all that kind of stuff, and the velocity. They don't really change their speed. It's just the force that they can produce, and it's a huge force. There's lots of alligator studies already looking at the force production, but it's interesting to think that those kinematic variables don't really change. They're just as fast as a hatchling as they are as an adult. That spawned all these different studies, and I'm going to quickly go through them because I do want to leave some time for some questions and that sort of thing. But you see, Micah Fern's curiosity spawned all these other types of studies. And so one of them dealt with, again, this idea that abiotic factors and biotic factors can influence a living thing. And so I was interested in looking at how the food availability, we know that as alligators, they in stomach content analysis, they're feeding on marsh rabbits, muskrats, but they're also feeding on fish and crayfish. So those two ideas, some are outside of the water and some are inside of the water. Emily Easter wanted to know, is there a difference in feeding above the water compared with below the water? And so we can also equate that to a less dense environment. Looking at viscosity, air is a lot less viscous than water. And so we can look at this first video here. And so we're taking, the slow motion camera, we're stopping, and, we're doing like different measurements, how fast they're going after the prey item, how wide their gape is, that sort of thing. And so we can compare that feeding above the water to feeding below the water. And you'll notice something pretty striking. So they're very clumsy. And if you didn't pay attention, watch their eye. As they start to strike underwater, they actually literally close their eyes, whereas above water, they're keeping their eyes open. That was one thing that we didn't know going into the study. They literally close their eyes. It kind of makes sense. They don't want to get their eyes wet, that sort of thing, but it does make them less successful. And so surprisingly, there was no difference in their feeding behaviors. Again, we're seeing that they're opening their mouths just as fast above water as they are below water. The muscles, at least from what we measured, aren't activated any differently. Again, above water, below water, they're equally the same here. And so that was another fascinating study that came out of that. Now, looking at some biotic components, so that was kind of looking at abiotic things. What about the influence of interspecific competition on alligator feeding behavior? Well, these guys, as you see from this picture, they're as hatchlings, they're not singular. They're going to be in a group. And so our question was, how does that influence the feeding behavior? And so this is a crude rendering of my photoshopping ability here is adding this alligator into this picture. But we actually had a focal individual that we did kinematic studies on. And then we added the same individual into a tank with a conspecific to see if that had an influence on their, maybe if they're competing for a limited resource, do they go faster? Do they change their kinematic feeding behavior? What we found was that's not the case. What you see in blue here is the control. That's the individual by himself. That's the focal individual by themselves, whereas the experimental group is with a conspecific. It's still the same individual, but now they're paired. And what you might see is how fast do they open their mouth, how fast is their attack velocity. The error bars say that their attack velocity is not really different. Their max gape is not different either. But looking at the attack duration, it looks like the whole event is shorter for those individuals that have a conspecific in the same tank. All right. But statistically, it wasn't different. And so we have to say they're not different, but there might be something going on. And so actually adding more replicates, we only had about 8 replicates in this study. And so we needed to get a couple more. And so here's kind of what's going on here. Again, we had the focal individual that was eating, and the other guy just kind of chilled. He didn't really go after it. He looked perturbed, though. He has his mouth open at that point. And so, but again, just spurring these ideas of different things. And we were going to continue that study, and then things changed. Darkness befell the land, and you guys know it as COVID. The COVID era began. And so that really shut down our ability to go travel, get hatchlings from our hatchery down in coastal Louisiana and bring them back to the lab. It changed a lot of things, and I'll talk a little bit about that. I was interested in looking at stress hormones in alligators and in fishes and in a lot of things. How do stress hormones influence their feeding ability? And so we partnered with the Bass Pro Shop in Memphis, Tennessee that had alligators. And so because we couldn't go get our own, we partnered with them. And so what you see here is actually they're getting water samples from the alligator display there, and that guy's guarding, make sure the other person doesn't get eaten. And the idea was you can actually sense some of these different types of hormones in water sources. There's new ways and novel ways to measure that. And so we set up a scenario that you see here in this middle picture where we had an alligator on one side, you can't really see here, it's a snout there, and then another one here, and chicken suspended in between. All right. And the idea was that if they were both competing for this one chicken piece, maybe the cortisol levels would be exuded into the water and we could measure it. That was the idea. Okay. It didn't quite work out, but here's a funny video. So it's kind of long, so I'm going to speed it up a little bit. So here's the chicken, here's the alligator looking interested. And then we find out really wasn't interested. It was actually interested in getting over to the other side to the other alligator. He just didn't want to be alone. So that was one of the drawbacks of this study. And so, but we're still kind of a work in progress on that. Another thing that came out of this COVID pandemic for me, again, the best laid plans of mice and men are just that. They're going to change. And so we had to find a project for this student, Samantha Jones. And so she was actually starting a novel project for me, which was looking at seagrasses. And we were able to partner with Palm Beach County, and they had a wealth of data about Lake Worth Lagoon and the seagrasses there. So what is a seagrass? It's basically a flowering plant that occurs in marine systems, and so the estuaries. And so so it has to be flowering, so it's not algae, right? And so it is different from that. And what you see here is a nice lawn of turtle grass, Thalassia testudinum. And so why is it important? It's a food source for many things. It also helps with nutrient cycling and sediment stabilization in these estuaries and provides a big nursing ground for a lot of invertebrate and vertebrate species alike. What's happening to it? We already know over the last 20 years, especially along the east coast of Florida, that this prized possession in this estuary, these seagrasses are being decimated, but nobody knows exactly why. It could be beach renourishment, it could be overgrowth of algae, but it could be input from the terrestrial system, from fertilizers or any number of things that are impacting its growth and stability in the East Coast. And so we partnered with Lake Worth Lagoon and the Palm Beach County Environmental Resources Management, and they gave us 12 years worth of data to look at maybe some of what correlates could be driving the rise and fall of these seagrasses in different locations. And so it's a big, long kind of explanation. And so this graphic representation, we built models to try to define what helps seagrasses increase and decrease in Lake Worth Lagoon. And that could be the physiochemical data, that's like temperature, dissolved oxygen, pH, those kind of things we talked about already. It could be spatial, so size of the different areas. It could be over month or year. That could be an explanatory variable, right? Because hurricanes come certain years and not others. Maybe that's why. And then just looking at the microalgae in the area, could that influence seagrass density? So lots of different models, 29 in fact, were developed for this analysis. And one model rose to the top. And that's what you see here. The best model explaining seagrass increase in density was year and microalgae abundance. Okay, so what does that look like practically then on a graphic here? So here along the x-axis, we have the years in the study. And then on the y-axis, you see mean BB score. Just know that that's a density measurement. So the higher the BB score, the more there is of that thing. So green is seagrasses, red are microalgae. And what you see from 2007 to 2012 is you have a pretty ebb and flow of seagrasses, but it's pretty prominent. And you also have an ebb and flow of macroalgae, sorry. But something happens in 2013. Dun, dun, dun. In 2013, seagrasses just plummet, and they continue to plummet. Maybe a little rebound here, but it's not looking good. But also plummeting are the macroalgae. And that's kind of curious because you could be like, well, macroalgae are the ones that are causing this, but they're not doing well either. And so we have these two dichotomous groups before 2013 and then 2013 and after. And that's a pretty stark contrast. And what was interesting about this is that 2013 seems like a pivotal year. There's lots of freshwater discharges that also other scientists and researchers were pointing out. Some of the environmental engineering groups had showcased that in 2013 and 2015 were big years for that freshwater discharges into the lagoon from terrestrial systems. And so that probably shifted the community structure in a big way. And lo and behold, next to the big sites that changed, Canal 17, C17, there in Broward County. And so, yeah, a big part of it could be this freshwater input. And so lots of people help out with these types of projects. And so we got to give them kudos for that and acknowledgments for that. And so this is actually the newest thing I've been working on, investigating the influence of turtle grass, Thalassia testudinum, communities throughout the Caribbean basin. And so my colleague Michael Skiebout has really helped me, and I want to continue these types of projects here at Nova Southeastern. And so again, a big shout out to all these supporting groups here, whether it's a research reserve in Rookery Bay or in Hobos Bay, or Department of Environmental Protection, Florida Department of Environmental Protection, that sort of thing. You can't do research in a vacuum. And so it's always good to make these contacts and give kudos where they need to be. And so again, I had lots of undergraduate help with this, and I'd like to continue it in graduate studies here at Nova Southeastern. And so basically, again, we're saying the same thing, that seagrasses along the east coast of Florida have plummeted. So why is that? But we also have areas that have thriving seagrass beds. And so that's the turtle grass is a really good one for that. It's a stable kind of seagrass. It provides all these ecosystem services, nursing grounds for things, nutrient cycling, sediment stabilization is a big one for turtle grass in general. And it's a food source for turtles and manatees, that sort of thing. And so what we know is that in the 3 locations you see outlined here, Charlotte Harbor Aquatic Preserve over in Punta Gorda, and then Rookery Bay in Naples, Florida, as well as Halmos Bay down over in Aguirre, Puerto Rico, along the southern portion of Puerto Rico. They have varying degrees of health of the turtle grass population. So in Charlotte Harbor, it's pretty stable. Puerto Rico has a thriving estuary full of turtle grass, but is very unstable at Rookery Bay. So the question that I have then is, why is that the case? Why are some stable and some are not? And so this study, it's continuing. We've collected 5 years worth of data, but I've also included the spatial scale because there could be local drivers at each and every one of these locations that maybe, if we're doing the modeling for, point to a driver of certain things. But if we increase that spatial scale, go from a local to a larger scale, so a regional scale, just looking at Southwest Florida or just looking at Puerto Rico in general, maybe that teases apart a different type of potential driver for those systems. And then the Caribbean basin as a whole, I'd actually like to add an East Coast site of seagrass for looking at Thalassia as well. And so there are lots of potential drivers. And again, it is based on scale, whether it's seasonal fluctuations, looking at the Caribbean scale, that's important, but that only shows up really in Puerto Rico at the annual fluctuation there, but it doesn't show up in Southwest Florida. Seasonality is not as important, okay? But if you look at the local scale, going back, seasonal fluctuation at Charlotte Harbor is important, where it's only annual fluctuation. So differences between one year and another at Rookery Bay. And so, and again, models are only as good as the inputs that you put into them. And so I'm always working to update. One of the big things I want to get into is looking at current models and overlay that in my models to see if currents are actually more important than these other things. And so other things I'd like to add, this is an invasive species that we can actually see in our data set. Our 6-year, 5-year, 6-year data set actually shows this invasive species of seagrass moving in to our sites. And so that interests me. Again, it's opportunistic, but I'm going to take it. I want to start adding that aspect into this study as well. And then we have sargassum, goes into the estuary just like it does here in Southeast Florida. We have sargassum comes into the beaches and makes a mess of things, but that also presents a problem for the estuaries there. And so again, I really have no special talent. I'm only passionately curious. I want to add to that. The important thing is not to stop questioning, because curiosity has its own reason for existing. And so I hope you guys stay curious, and I have time for questions. Oh, and this is, I like to jab Tennessee football anytime I get. And so anytime they play Florida Gators, I usually post this on my social media. Because Tennessee usually loses in these cases. So, but thank you guys again for listening, and I'll take any questions.
Awesome, Dr. Kerfoot. Thank you very much for that lecture. So if anybody does have any questions, please feel free to put them in the chat, and we can answer a couple questions. I do want to make sure that we, are cognizant of everybody's time. We're running a little bit over than normal. That's totally fine. But if anyone does have questions, please throw them in the chat for us so we can answer them. If you do have other questions that you think of later, please feel free to contact any of my staff, and we can definitely forward it on over to Dr. Kerfoot to answer for you. So while we are waiting for if any questions do end up coming in, I always like to ask, obviously, Dr. Kerfoot, you are new to NSU, which has been very exciting getting to know you over the last couple of months since you just started with us. What's one of the things that attracted you to want to come work at NSU? If you didn't already discuss that specifically.
No, I didn't. Actually, thank you for asking that question. I think there comes a time in a researcher's life is like, okay, what do I really want to be doing? And I think, again, I have to blame it on COVID. I was sitting there teaching online classes, and I hate teaching online classes. And I said, where would I like to be? and where would I like to be teaching and doing research? And I was already starting to do research with seagrasses, and I was like, man, I'm going to just take the jump and try to get back down to South Florida. And that's where that's where I ended up. And not that, I was doing pretty well doing the seagrass research in Tennessee, West Tennessee, but it was getting kind of logistically tough. So why not be closer to the action? And so, but what attracted me to NSU specifically, you already have high-powered researchers here doing numbers of different things, from coral restoration to coral propagation, to looking at avian parasites and all the things in between. And so this is a happening place, NSU is, and I wanted to come be part of that excitement as well.
Awesome. And I actually do have many questions, but I'll limit myself to just try and do one. No, just because you did mention, the sargassum, which is obviously I know kind of towards the end of your lecture, but that is also something that we do see and smell quite frequently. Is there any type of work being done to make that product useful in some capacity?
Yes, I know of one specific example in Puerto Rico. Because going there an awful lot, we get to know the estuary managers, and they're looking at ways to. They've been harvesting the sargassum. They've been kind of spraying it down because it does have the saltwater component, but trying to utilize it as fertilizer. So they're doing some micro experiments on some of the agriculture there to see if it would be beneficial to use as some sort of fertilizing source. But it does take money and time to actually clean it off in the sense of the salt water. But yeah, so those are opportunities, and it's actually interesting. The sargassum that's coming towards us is actually coming off the coast of Colombia, and so it's kind of fascinating that certain years are heavier than others, but it seems more prominent these last 2 years, 2 to 3 years. I don't know, at least in Puerto Rico, I think it's probably been happening the same way down here. And so are there other ways to use it? People are thinking about it because it is a problem. How can we solve the problem, getting it off our beaches, but also make money from it too? Yeah.
Absolutely. And then since our guests are being shy, I will have one more question before I kind of let you go. I'm sure you got plenty to do as well. You mentioned obviously the invasive species that have been of seagrass that have been kind of working their way up the coast. Now, traditionally in Florida, invasive species are incredibly detrimental to the local wildlife. Is that something that's similar, or are turtles just kind of going from the native grass to the invasive one with no problem?
That's great question. And what I recently found out, there was actually the same invasive species that I saw in Puerto Rico, they found in Biscayne Bay. And so that's alarming. And let's take a step back. Yeah, it is alarming. You say invasive species. Invasive itself has some sort of negative connotation. But your question really drives at, are they being detrimental? Now in Puerto Rico, they've done some autopsies on manatees that wash up dead, and they have found this seagrass, this invasive seagrass species in their stomachs. It's not the seagrass though killing them.
Right.
You can maybe draw that conclusion. But what's happening is they seem to be actually selecting for that. Who knows why they might prefer it? Maybe it tastes better. Maybe their body just says it's energetically favorable. But they are targeting it. But that's also what's helping them move, because this invasive grass, manatees are sloppy eaters, and they can fragment it, and that fragment of this seagrass can just get in the current and move very fast distantly. Whereas the native grasses have a harder time doing that. They've actually adapted, like turtle grass, to rooting their vegetation into the substrate heavily. And so if they get their top blades chopped off, that's okay. It's their growth mechanisms are closer to the substrate. So unless we have manatees going all the way and rooting it out, you're not going to get that same propagation mechanism. And so that's helped this invasive species do well. Whether it's detrimental, we know it could be detrimental for the native seagrass because it overcrowds it. Is it
Okay.
detrimental for manatees? Maybe not.
Okay.
Good question.
Awesome. Well, Dr. Jarrah, thank you so much, like I said, for being here. My staff as well. Thank you guys as always. We appreciate any support we can give. And for those of you that are watching tonight and online later, thank you so much for being here. It's been a pleasure to get to, listen to another virtual lecture series run by our Department of Marine and Environmental Sciences here at Nova Southeastern University. We will see you all hopefully coming up in another month. We do have our virtual open house for the department coming up, so stay tuned to an email coming from us about more of that and login information. We'll go over more of the admission side and some more information about the department labs and more details specifically about NSU. So thank you all once again for being here. I hope you have a great night, a great start to your week, and I will hopefully see you all on campus soon. Take care, everyone. Bye-bye.
Thank you.